Alrighty. We're looking at a particular cell. What is this cell called? A rod. Where do you find it? In your eyeball. All right. What does the word chromophore mean? Yeah, it's where the light, the actual photons, are absorbed. What is the chromophore in a rod cell? Rhodopsin. It is a protein and a vitamin. Where is it located, the rhodopsin? In what? The iris? No. In the retina, yes, but where in the retina? Where in the cell? The cells make up the retina. Okay, I don't really care. It is located in the membrane of the discs. And you see where those discs are? And they're embedded all in those membranes. Okay? Lots and lots. Sorry. All right.
It's a good thing we had touch.
So now we know where we are with what we learned last time. We're in those discs embedded in the membrane. Remember, all the signaling has been happening, to get something across membranes. All right. So the rhodopsin, once it absorbs light, what is the transition?
Cis to trans.
Cis to trans, very large movement, and then a conformational change. When an enzyme or when a hormone binds a receptor, you have a binding in the pocket and you have changes in the shape or conformational change. Here, the light is going to cause a conformational change. The rhodopsin is a chromophore. It is also a mimic of the receptor. It's receiving the light. All right. In the rod, in the cells of the rod, around the membrane itself that houses the entire cell, there are ion-specific channels, and they are open in the dark. So that means ions are flowing from outside the cell inside the cell, and the ion is sodium, and sodium is coming inside the cell. Okay, this is in the dark. So we are going to have a response when light shines, right? And so if it is flowing in the dark, what do you suspect will happen when the light shines?
Shuts the channel.
Shut the channel, and I'll stop that flow. Okay. Ultimately, you just now have a preview of what's going to happen. Let's see how that happens. All right. So in light, the absorption of rhodopsin causes the closure of the ion-specific channels. One photon will block about 1 million sodiums coming in, and when you do that, you get what is called hyperpolarization. All right. Now, what is depolarization? We use this in a nerve cell. What does that mean, depolarization? I depolarize the cell. I have an action potential. I have a nerve impulse. But what does that actually physically mean? Oh my goodness. Yeah, it's something about the ions. Normally in a cell, it is negative on the inside because there's more sodiums on the outside than on the inside. And when you open up the channel, the sodiums flow in, potassium flow out. And what happens in a depolarized cell, the inside of the cell becomes more positive than what it was in its resting stop. Here, in this case, we become hyperpolarized. Sodiums in the dark are coming in, and then all of a sudden I close the channels. And so what I do is I become more negative on the inside than normal. And that's a hyperpolarization. Both of which are an electrical change across the membrane. Okay? And it will sense this in this entire cell, which then will allow the neurotransmitter vesicles to merge with the cell membrane and release a nerve impulse. Okay, I'll give you an overview and then we will go step by step.
In the retina of the eye are In the outer segment of the rods are stacks of membranous discs, and embedded in the disc membrane is the photoreceptor protein rhodopsin. Bound within rhodopsin is the light-sensitive molecule retinal. In the outer membrane of the rod are CGMP-gated sodium channels. While CGMP is bound, they remain open and sodium can enter the rod, depolarizing the cell membrane. When a photon of light strikes rhodopsin, retinal isomerization initiates rhodopsin activation. Active rhodopsin binds transducin, a trimeric G protein. G-alpha moves away to another membrane-bound protein, CGMP phosphodiesterase, or PDE. When G-alpha binds, PDE converts the available supply of CGMP to its non-cyclic form, 5'-GMP. As CGMP dissociates from sodium channels and is converted to 5'-GMP, the sodium channels close. When sodium can no longer flow into the cell, the membrane hyperpolarizes, sending a signal down the cell to its synaptic terminal, where the signal begins its journey to the visual centers in the brain.
All right. So we've seen cyclic GMP as a second messenger before. Okay. We've seen a G protein, although in this case we give it a name, transducin. Okay. We've seen the enzyme phosphodiesterase before. It breaks down cyclic intermediates. And so we've seen all of these players, just not in this combination. So the channel will close in the light, okay? And when that happens, what you have to do is get the concentration of cyclic GMP concentrations reduced so that those channels close. And it does this in response to the absorption of rhodopsin. Molecules involved, rhodopsin itself, transducin, you can write G protein next to that, and then phosphodiesterase. Transducin, again, has an alpha subunit, which is the catalytic, and the beta gamma subunit, which is regulatory. The alpha subunit, when it is bound by gamma and beta and has GDP, the diphosphate, it is inactive. But once rhodopsin has been stimulated, the alpha subunit will release the beta gamma, will release the diphosphate, and pick up a triphosphate, becoming activated. The PDE, phosphodiesterase, actually is a 3-subunit protein. Alpha, beta are the catalytic, and the gammas are the regulatory. It breaks down, and its job is to take cyclic, in this case, it's specified for GMP, and break it down to 5' GMP. Signaling molecule, not a signaling molecule. Okay. And this is the reaction that it will catalyze. It will break the phospho, and you can see why it's called a phosphodiester. An ester bond with the double bonded, and then a ester with the double bonds of phosphodiester, and it'll break it down. So, light will come in, let there be light, and it will cause a conformational change in rhodopsin, and the star means that it is photoexcited, and there is a physical change. That physical change allows the transducin to bind to it and initiate the removal of the beta gamma and the exchange of the diphosphate for the triphosphate. And now I have the GTP that is roaming around. The GTP then can float in the membrane to PDE. It will bind to PDE in such a way that it actually removes the regulatory subunits, creating an active PDE, which then takes lots of cyclic GMP and create lots of 5' GMP. Once the concentration of cyclic GMP goes down far enough, it will not effectively bind to the sodium channel, and when it is not bound, the sodium channel closes. And so, rhodopsin gets photoexcited. It will then stimulate the alpha subunit to become active, which then will bind to PDE to make it active. PDE hydrolyzes cyclic GMP, and the 5' cannot bind to the ion-specific channel, and it causes it to close. The cellular response then is depolarization and signaling of a nerve signal to the nerves. All right. So, how in the world do you stop this? Please stop. Close your eyes. Go into the dark. Okay. The main players. I have to stop the photoexcited rhodopsin. I have to make transducin inactive. I have to make PDE inactive, and then I have to get the cyclic GMP concentrations up to normal. Okay. We will then see a new enzyme in the mix. The rhodopsin is phosphorylated once it is, and only if it is, in the excited state. Okay, and it phosphorylates it. Once it's phosphorylated, a protein will come and bind to it called arrestin. All right. We have seen in multiple cases that the receptor itself, once it changes its shape and is able to upregulate a G protein, also changes its shape somewhere else to open up a place to be phosphorylated. And once it's phosphorylated, a protein binds to it. All of them called arrestin. Some of them have a beta, some of them have other desiccants in front of them, but it's essentially a protein called arrestin to stop. At this point in time, it is still photoexcited, but it will not upregulate any more G proteins. And so what we have to do is we have to take their receptor, the rhodopsin, allow the retinol to be released, converted to retinol, re-isomerized, put all back in. We talked about that particular process. The transducin itself, all G proteins, the alpha subunits have a GTPase activity. It is relatively slow, but as soon as it breaks down the GTP into the diphosphate, it stops. Once it stops, that means it unbinds from PDE. PDE was only active if it's bound to the alpha subunit that has a GTP bound to it. So, in essence, both of them shut down. But at that moment in time, we have killed all of the cyclic GMP. They've made it all the way back to 5'. I have to get that particular concentration up. Cyclic GMP concentrations are regulated by calcium. Normally, calcium is at about 500 nanomolar inside the eye, which is much higher than normal other cells. When the light closes, all right, calcium, when the light closes here, calcium also stops coming into the cell. Okay, and it reduces to about 50 nanomolar. Once the calcium goes down, it stimulates guanylate cyclase, which we've seen inside before, which takes ATP, and this enzyme will convert it to, won't take ATP. It will take GTP and convert it to cyclic GMP. And the concentrations will go up. So as the calcium concentrations go down, the cyclic GMP concentrations go up until it effectively binds to the channel, opens up, everything starts flowing through once more. Again, a new enzyme as part of the sequence, but it's an enzyme we've seen before, just in a different order. All right. Now the rods detect black and white. Okay. But we have cones that can detect color. Now, what happens is, to detect color, we actually have to detect 3 different versions of color, RGB. And so you would think that the organic molecule, if we add or take away double bonds, you can manipulate where the absorption, the maximal absorption is for each one of those. Here is the problem. The cones and the rods, all of the receptors still use retinal. Okay? The same organic molecule. If you put that organic molecule in a UV vis, they will all absorb at the exact same maximum. Okay? But when you put them inside the proteins, different proteins that are versions of rhodopsin, you get shifts in where they absorb. Why in the world would the same organic molecule have different shifts? The reason why the organic molecule can absorb is that conjugated double system, conjugated double bonds. Okay? When it binds inside a protein, the protein side chains interact with that organic molecule. And you can have interactions of the R groups that might retard or help expand the ease at which the double bonds are conjugating. And so the pocket influences that organic molecule to change its absorption just slightly. And so we can get absorptions of 4 different maximums depending upon what version of the opsin protein that you're bound to. Okay, and there are different versions for each of the different colors. They're homologs. Okay, signaling in terms of color tends to be this. How much signal for each one of the 3s are being detected at any one time? And so different intensities of the 3 combinations will provide different colors perception. All right, so we've just done eyes. We are now going to look at what? We're going to look at smells, okay? And so the signaling molecule in this case is not a hormone, it's not light, it's an odorant, okay? And if you've ever walked into a middle school, there are lots and lots of odorants because someone has done a very good job in advertising that Axe will attract exactly what these male students want, right? So instead of putting it on, they bathe in it, all right? Or first week of Lee, you might be able to tell a freshman girl if she's carrying a purse and maybe wearing way too much perfume and all made up. Guys are all dressed up first week, and then after that, just roll out of bed in pajamas for both sexes. Okay, so odorants are being given out all over the place, all the time. All right. Now the odorants are going to be bound by an OR, an odorant receptor, okay. Or there can be binding proteins, all right, that can bind the odorant before it binds to the receptor. Now, what you can't see in this projection, but I think you can see in the printed picture, is that these odorants get caught in the mucus layer, okay? And so they have to diffuse through that. The binding proteins and the receptors are in the mucus layer themselves. Not everyone smells things the same. The amount of receptors you have and the versions of the binding proteins and the receptors you have are different from person to person. And so people smell things differently, or some people can't smell anything. After being in an organic synthetic lab for several years, I smell very little. Okay? I just totally burned out with all of those organic molecules. All right. So a signal comes in, some sort of odorant, binds to the receptor directly or through a binding protein. And check this out. We get a protein that has an alpha, beta, and a gamma subunit. The alpha, the theta and gamma are regulatory. The alpha is stimulatory, and it exchanges a GTP out, picks up a GTP. What is this kind of protein? A G protein. The alpha subunit upregulates what's AC, adenylate cyclase. ATP is converted into cyclic AMP. I told you need to know up to that. This is some other pathway. We saw cyclic AMP in another pathway upregulate pKa. Okay, what is it going to do here? What does that mean? Well, what does this mean? Stop. So this means go. Good. All right. It is a channel opens. What kind of channel is it? Calcium channel. Okay, the calcium channel opens, and then the calcium channel comes over to a what channel? A chlorine channel. Chlorine channel opens up, and chlorines flow out. Is this cell polarized or depolarized? Depolarized. Negatives flow out, so it becomes more positive on the inside. Okay. All right. You can see to stop this, you've got to get calcium down, because once calcium is down, this will close. I've got to pump chlorides back in. And you can see this calcium channel goes and stimulates the response and starts to turn itself off because it will close itself. How do I break down cyclic AMP? I use what protein? Phosphodiesterase. Goes back down. How does this alpha subunit shut down? It breaks its own GTP by a GTPase activity and reassociates. Once this is broken to GTP, this stops working, so I don't make any more. Okay. How do I get rid of the odorant? I run away from it. Okay. Most odorants have to be oxidized by other enzymes to actually break them down. Okay. They just don't diffuse away too well. And the receptor can also be phosphorylated and bound to an arrestin to stop it, even in the presence of the odor. Okay? You can see, you can smell. Let's taste. Okay? We're going to have an example of the sweet receptor. There's a debate about how many receptors there are. Before, way back when, it was bitter, sour, sweet, and salty. We've got a couple other receptors that are being purported as being there. A fat receptor, possibly a carbonation receptor, and then one I can't pronounce. And so, all right. So a sweet receptor. All right, we eat it. It by sweet. Taste it. Oh, this is great. It's a taste. An odorate. A taste. All right. Binds to a sweet receptor, and a G protein. But now I have a name for it. So instead of transducin, we got gusducin. Woo! All right. And so stimulating a G protein, the alpha subunit, adenylate cyclase again, to make cyclic AMP. That seems to be a reoccurring theme. Now, what in the world does cyclic AMP do? Oh, check this out. Upregulates pKa. We know how that works. Then pKa does what? Phosphorylates a channel and causing it to close. Do you see that? It goes from an open before phosphate to a close after phosphate. All right. Is this a hyperpolarization or a depolarization? It becomes more closes. Normally it is flowing out. Closes it, more positives are on the inside than the beginning. And that is a depolarization, correct? Okay, and then that will send off a signal. All right, you can see, you can smell, you can taste, and that's all we're going to do. Okay. Now, in this case, the commonality of the G protein coupled. All right, in mammalians, we have 30 to 40,000 genes. There are thousands of these proteins that are in these pathways. It is a large fraction, upwards of 10% of all proteins that are produced in the genome are in these pathways. Okay? And so most of the binding events cause a receptor change in a three-dimensional shape, activation of some sort of second messenger, which then interacts with a channel or a kinase. We looked at the sweet, okay, and what happened. We looked at the odorants, but in this pathway that affected cyclic GMP, there is another way that affects IP3 formation. We looked at light for rhodopsin. We've looked at epinephrine and upregulation of pKa. But there are other things like vasopressin, which hits the receptor, and the G protein in this case is a GI protein, whereas this was a GS protein. Gets to adenylate cyclase, which then lowers the concentration of cyclic AMP, and pKa function goes down. So we can have opposite. It doesn't always have to stimulate, it can inhibit. But there are lots and lots of these. A very large fraction of all the proteins in our genome fall into this category. All right, there are some bad diseases associated with this that actually are effective in the G proteins, both cholera and whooping cough. Cholera inhibits the GTPase activity. And so in that case, it will make the alpha subunit always or constitutively active. This is not a good thing. And so we always need to have a biological system where we turn it on and turn it off. Whooping cough inhibits the GI, and it stops the exchange of GDP for GTP. And so when the signal comes in to downregulate a process, the whooping cough will not allow us to downregulate. And so both of these, cholera and whooping cough, will disrupt the normal signaling pathway. They do this by modifying covalently the actual protein itself. And so we'll take one of the R groups and it will add on a, in this case, a portion of the electron carrier, and that will disrupt the binding events and how it all works. So diseases that can be associated directly with G protein disruption. Now, we talked about protein kinases being activated, and so we talked about protein kinase A goes and finds its target, and protein kinase C goes and finds its target. We also talked about protein kinase G and protein kinase B and lots of protein kinases. Now, the animations even showed you they floated around and found their targets. But remember I said that most of the time our regulatory events are joined together physically as well as the stepwise process. And so enzymes that bring the protein kinase and the target together are scaffolding proteins that keep that whole mix together. In the case of pKas, these scaffolding proteins are called AKAPs, the A kinase anchoring proteins. And then somebody got really creative with protein kinase Cs. You have RACs and you have PICs. RACs are receptors for activated C kinase. They really had to force that, but it actually makes a good acronym. And proteins that interact with C kinase. And so, but what a RAC or a PIC is, it sits here, it binds to protein kinase C, and it binds to protein kinase C's target to make sure that they are right next to each other. And so it is an organizational event that organizes those proteins, which is now important when we think about overall regulation of a process. Not only are the protein kinases there, the phosphatases that are associated are also bound. And so you have a kinase to activate, you have a phosphatase to deactivate. Or you have a kinase to deactivate, the phosphatase there is to activate. And they're all bound together. Steroid hormones work a little bit differently, okay, in that they are actually a lot more slow in their response. When you hit a receptor and you get a G protein and you upregulate adenylate cyclase, you get all of these exponential attenuations or amplifications. And so one response can get thousands of responses from one signal. The steroid hormones work a little bit differently. They actually will diffuse all the way through the membrane, all the way into the nucleus, and bind to the receptor there. Now that receptor, steroid receptor, we talk about it in a different light. We don't talk about it as being receptor, but we talk about it as being an activated transcription factor. So the receptors become transcription factors upon binding of the steroid hormones, which then causes the proteins to be translated from the DNA itself. Epinephrine, very quick in its response, seconds to have a cellular response. A hormone release may take days for it to actually fully engage because of the differences in pathways. Here, I've got to go all the way to the DNA, upregulate the proteins. The proteins then have to get into a high enough concentration, then they become active, then it becomes fully engaged. And so hormones, hours to days. Other receptors can be seconds to minutes in terms of their responses. Steroid hormones. Two examples, tamoxifen and UR486. Tamoxifen actually is a competitive inhibitor, and tamoxifen competes with estrogen. Does anybody know what tamoxifen is associated with? Has anybody had experience or a family member? It's the treatment of breast cancer. And so what this particular drug binds, it's a competitive inhibitor. It'll bind in the same place that estrogen will, but it will not cause the protein or the signaling pathway. Tamoxifen, wonderful drug, but there was a very large controversy when it was started to be used. Before we found a synthetic pathway for tamoxifen, it was only found in the tree bark of a endangered species. And so, do we harvest the tree bark and kill the tree to extract the natural product as a drug and give it to someone to save their life, or do we protect the trees and not save their life?
Plant more trees.
Plant more trees. Takes a long time for them to grow, and people love trees. And so, once we did find a synthetic pathway, that issue went away, other than, I want a natural product. What is a natural product? The product that's found in nature. If I synthesize it and it looks the same, it's a natural product, made synthetically. All right, there, I'm off my soapbox. All right, RU486. Okay, this compound right here was one of the compounds that my major professor when I went to grad school was about to look at in terms of the synthetic pathway. And so my project would have been to work on this. And I had a problem with that. An ethical dilemma about thinking about that. Now, the state of Kentucky's legislature made a law that said no university in the Kentucky system could work on this particular drug, and so that solved my issue, even though my professor received a grant to do this. RU486 is now sold in stores, but not under the RU486 name. It's actually sold under the day after pill. Okay, and so again, this competes with the progesterone, blocks the hormone action that allows the implantation of the fertilized ovum. And so that would have been graduated from leave, and I graduated in May, started my research in May. But during my interview process, he told me that this was going to be, I went up there thinking I would have had to have a real long conversation and maybe change my major professor, but between the time I interviewed and the time I graduated, the Kentucky legislature helped me out. So, go God. All right. All right. All right, we're going to look at one more pathway for signaling. Okay, and it's going to be in terms of the cell cycle. Okay, how many times have you learned the cell cycle? Did anybody see this in high school biology? Yeah. Biology 109? Yeah. Oh, you've seen the cell cycle, right? So I'm going to give you a close overview. But then what I'm going to teach you is how the cell internally sends the signal to cause changes from one cell cycle to the next.
During most of the cell cycle, the nucleus is in interphase, represented here by the 3 green segments. During G1, the first gap phase, metabolism proceeds and the cell may grow. The S, or synthesis phase, is when DNA replicates. The S phase is followed by G2, a second gap phase. In the M phase, the nucleus divides. Then the cell cycle begins again.
Okay, this is what we know, right? Got this down. All right. What we're going to do is we're going to let you know that the signaling to go between those cycles when the cell actually transitions, it is regulated by 2 different protein classes, one called cyclins and the other called CDK, or cyclin-dependent protein kinases. There are 10 different classes of cyclins and about 8 different classes of kinases. Depending upon what is present in the cell at what time, the different combinations will produce different events. So if you see that this slide down here, of this picture, the combination of the version cyclin E and the CD kinase 2, CDK, is right between the gap phase 1 and the S phase. And then the concentrations will go up and down to cause changes in those different pathways, or those 4 different phases.
Cyclins are proteins that regulate the cell cycle. The CDKs are cyclin-dependent proteins that must complex with cyclins in order to act. S-cyclin accumulates and binds CDK, forming the M-phase promoting factor, MPF. MPF activates other enzymes that initiate the signal to synthesize DNA. Now the S-cyclin degrades, and another cyclin, M-cyclin, accumulates. M-cyclin binds CDK, forming a new MPF that activates still other enzymes, initiating the mitosis signal. Finally, the M-cyclin degrades, so that the cycle can start again.
Now, what's important about this, okay, is when this particular protein is bound by others, okay, this kinase is bound by one, it has a target for X. When it's bound to another cyclin, its target is Y. And we talk about proteins as being very specific. When I bind to a cyclin, I actually change its specificity pocket from one entity to another. And so this is one of the first examples in which you're going to see an enzyme change its specificity depending upon what it's complex to. All right. Oh my goodness. Again, when you see these things, it's scary, right? All right. It's a cycle, so it starts and goes around. So where do you want to start at? 1. Okay, we can do that. All right. So at 1, here I have a cyclin-dependent kinase. All right, I didn't tell you what version, but this will work for all of them. And it's not active. Okay. Upon some sort of signal, one of the cyclins starts coming up in concentration. Okay, building in concentration. And it will bind to the CDK. Okay. And so I now have a complex. What occurs here is that the complex will allow this to be phosphorylated once. Okay. Once that is phosphorylated once, it brings in and it actually allows a second phosphorylated state. Okay. We're almost ready. Once the second portion is phosphorylated, it gets dephosphorylated into the active complex. All right. 3 things occur. As in the last slide, the first thing that occurs is cellular response. Okay? It starts causing the phases to change. Right? The second thing, 2. This will work on a phosphatase. It'll phosphorylate the phosphatase, which will then cause more of the inactive or partially active into the fully active. This number 2 is a feed forward, meaning that I want to work faster and faster and faster and faster. Third thing, turns itself on in number 2, and it begins the process to turn itself off in number 3. What it will do is it will phosphorylate the destruction box recognizing protein from an inactive state to an active state. This destruction box recognizing protein will then cause the ubiquination of the cyclin. Have you ever heard of the protein ubiquitin? Okay. Ubiquitin is a small protein that is ubiquitous. Does anybody know what ubiquitous means? It's everywhere. It's everywhere. Okay, so what this does, normally ubiquitin will add to proteins slowly over time. It is like an aging protein. The more ubiquitins get added, once you get lots of them added, it signals the cell that this protein has been around long enough, send it for degradation or recycling. In this case, the cyclin hasn't been here very long, but there's a protein that is upregulated to start adding ubiquitins onto that protein, the cyclin. And so it will add it very, very quickly, making it age quickly. And once you have so many ubiquitins on any one protein, other processes in the cell take that protein and send it to the lysozyme to be broken down and recycled. Once the cyclin concentration is broken, the cyclin is broken down, I will return to the inactive state. Okay? So, the cyclin-dependent kinase will phosphorylate things to cause changes in the cellular response. It will turn itself on by upregulating the phosphatase to cause it to become faster and faster. Okay? You only get to a speed in which you convert all of this very quickly, then it's all active. The slower place is the destruction box recognizing protein, which causes the cyclin to be tagged for destruction. Once it's tagged for destruction, it'll go on to the next. It'll downregulate its activity. And so it has 3 main things that it does.